Okay, I think it's about time. So please welcome everybody to this new meeting in ReMicS. And today it's Knut Rudi who will talk about mother-to-child transmission of gut bacteria. So please, Knut.
Thank you very much for the kind introduction, and also thank you very much for being invited to give this talk. And I'm going to give a talk about one of my favorite topics, and that's how bacteria are transmitted from mother to child, and not only during delivery, but during the first years of life. So I think I'll just go like here. So why is this important? And it's because it's really like the first bacteria that colonizes are crucial for our lifelong health. So during the first couple of years of life, if you're not exposed to the right bacteria, then you may develop diseases such as allergy or other types of inflammatory diseases. And a particularly important aspect is to what extent do delivery contribute to the bacteria that you get, that the child will inherit from the mother. And probably aware of, there is a lot of discussion on whether or not one should do seeding of infants that are delivered by cesarean section. So in some countries there is a very high percentage of cesarean section, and that's really breaking the natural way of delivering bacteria to the infants. So there's a lot of discussion now on whether or not one should do seeding of bacteria, for instance by vaginal seeding, or perhaps also fecal seeding, and how this should be done. But in order to have the information basis for this kind of quite what to say, this is a quite drastic interventions. You need knowledge about the natural what to say, how bacteria naturally transmitted from mother to child. I just want to show you this picture. I don't know if all of you have seen this before. This is very quite old research, and it's the research conducted partly by Knut Rudi, who's a Norwegian. And I don't know if I think it was not this year but last year. These people were very close to get the Nobel Prize for this discovery. And it was Jeffrey Gordon. I think that was really like the one that was like one of the top candidates for the Nobel Prize in medicine. And the discovery that was back in 1996, where they saw that there was some specific bacteria. When you introduced this bacteria to bacterial-free mice, these bacteria were able to induce their own nutrient production from the gut epithelial cells. So the bacteria could induce glucose production from gut epithelial cells, and these bacteria are able to utilize glucose as a nutrient. And that was really like the first example on potential positive selection of gut bacteria. And the bacteria, the genus is the Bacteroides genus that had this kind of property of inducing its own food in the gut, and this was the mice infant, but most likely the same things happens in humans. Illustrating in a way that most likely the first colonizers would be important and for in a way the lifelong health of the host. This is a really major discovery, and this was partly done by the Norwegian Knut Rudi. Not let's say, I think it's 4 or 5 years ago. There was a so-called discovery that one could find in a way bacteria in the placenta prior to delivery. And I know bacteria that partially should have similarity with bacteria living in the oral cavity. And the authors claimed that there could be a mechanism of in utero colonization, so that we are colonized by bacteria before we are delivered. And there's been a lot of fuss about that, potential mechanisms on how this bacteria can be transmitted to the fetus, and the potential role of these bacteria prior to delivery, stating that in a way we could be colonized by bacteria before we are delivered. We did an experiment a couple of years ago, and that was through collaboration. We did not do that. It was in the Preventadal in a way. So it was within the Preventadal study, which is a very large study of mother and children with the aim of intervention towards allergy. But a side project here was looking into the effect of cesarean section on the bacterial colonization of the infants, and looking into how cesarean section would be affect in a way the amount of bacteria in amniotic fluid. So the genius thing they did was to look at cesarean section whether in the case when the amniotic membrane was intact, and in the case when the amniotic membrane was ruptured. And really, what we saw, and I think this is quite extraordinary results and very important results, that we saw that if the amniotic membrane was intact and the infants were delivered by cesarean section, there was very little bacteria in the amniotic fluid. But if the children were delivered and the amniotic membrane was ruptured before the cesarean section, then we found quite a lot of bacteria in the amniotic fluid. And at least I believe that bacteria, the bacteria have no role in the fetus before delivery. And if there are bacteria, there could be bacteria, of course. But my view would then be that this would be pathogens, so that in a way there have been pathogenic bacteria that had in a way crossed in a way the barrier of the amniotic membrane, and that would cause disease of the infant. But you also have to take, there's a lot of studies studying the effect of cesarean section. It does not take into account whether or not the membrane, amniotic membrane is ruptured before delivery. And if you do not take that into account, that could be some kind of big mistakes then in thinking that in a way that the infants have been colonized by bacteria before delivery. In some respect they are, because they will be colonized when the amniotic membrane is ruptured, but that will be maybe just some hours before delivery. But some people would have said that in a way, okay, you can see that there's a bacterial colonization even for infants that are delivered by cesarean section. But we would claim then that this would be due to the rupture of the membrane, and that they have not control for this. Another very interesting aspect is in a way the PCR contamination controls, because in this study they had a control where they took the amniotic fluid into some kind of metal bowl that would normally be used in a way to collecting amniotic fluid. And by exposure to this metal bowl, we could find a lot of bacteria in the amniotic fluid. And then these bacteria do not actually originate from the amniotic fluid, but they originate probably from the bowl that is used. And that's in a way, it's really like you have to be very cautious when you are working with a low copy number, and when you're working with bacteria, when you have a very low bacterial load. And in this case, we did use digital droplet PCR for quantification, because that's a really good technique for quantifying a low amount of bacteria. Another study that we done some years ago, we were looking into association, looking at the bacterial OTUs, and these are like bacterial species defined based on the 16S RNA gene, and looking into the overlap between this OTUs between mothers and the children. So you can see that in a way, the first graph here is in a way the number of OTUs we find, or the number of bacterial species that we find in mothers at pregnancy and 3 months after delivery. So you can see here there's about 130 OTUs in average in mothers. And if you can look here now, you can see the 10-day children, we are down at 20 OTUs. There's a very small fraction actually that, at least at the OTU level, that are detected in the 10 days children. And then you can see that the number of OTUs are gradually increasing to 3 months, a little bit, 1 year, a little bit more, and 2 years. Then you are maybe about 2/3 of the OTUs of species that you have in the mothers. So I would guess that in about 4 or 5 years, in a way, you would have recruited all the bacteria that are like, so the infant or the child would have recruited all the bacteria that are present in the mothers. If you look in the bacterial classes, in a way, which the taxonomy of the bacteria, you can see that this big black thing here is the Clostridia. And it's really like the most diverse, predominant class. So these are the number of OTUs belonging to this class. So you can see that most of the OTUs in mothers or the bacterial species belong to Clostridia. And very interesting part here is this blue one, because this you can see this blue part, which is the Bacteroidia, or where you have the Bacteroides genus within. You can see this is this size in mothers, in infants, it's the same size. In 1-year-old children, it's the same size, and in 2-year-old children. So this in a way class does not seem to be age dependent. But if you look at the Clostridia, you can see that here the infancy is very low, and you see it gradually increases. And the hypothesis for this is that the Clostridia can be spore formers. So that in a way, although they are strictly anaerobic, they will not survive outside the host. They can probably generate spores that will survive and germinate when they get in the gut into favorable conditions, and then that we can recruit these bacteria when we are older. The blue one here is the Bacteroidia. These are strictly anaerobic, non-spore formers. So these would be like the perfect candidate for direct transmission, because they do not have, in a way, the possibility to have this kind of spore form, and will be much more difficult to transmit independent of delivery. Please stop me if there is some kind of questions or something, and if you want to clarify something underway. So there's no problem if you stop me in my presentation. And here's, in a way, looking into the overlap. So now we are looking into this, and this is what is called real, in a way, is what we observed. And a real, oh, sorry, this is a little bit like, this is real, it's mother-child pairs. So now we are looking into the child, and we're looking into overlap with the child's own mother. Okay, and then you have the number of OTUs, it's the red circle here in the mother, and the blue circle is the child, and then the overlap intersection is how many OTUs the mother share with her own child. And you can see that at 10 days there's only an average 8 OTUs, and then you can see at 3 months it's still is 8, but you can see that the child's increase a little bit, and then you can go to 1 year, you can see that, you know, the child's circle increases and the overlap increases, and then it goes at 2 years, the overlap increases more, and, you know, the child's circle increases. This is the child's own mother. But if you just take a random mother, whatever mother in the cohort, then you can still see that there is no difference. So on average, in a way, it seems like, in a way, the infants are inheriting the bacteria from the general population. So most of the bacteria, and this probably mostly reflects, in a way, the Clostridia, the spore forms. So the most of the bacteria that we have in our gut are recruited from the adult population, but not directly from the mother. They are inherited later from the general population. But we are also interested, I will come back to these bacteria that are directly mother-child transmitted, because there are also bacteria, but these are not the dominating. So if you're looking overall, all the bacteria in the infant, most of them will not be directly shared between mother and child. Most of them will be shared by the general population. here, and now we dig into the mother-child associated bacteria. In this, we use some time on the slider, so you can look into the bar here, this orange bar. In this case, we are gone to species level. So this is a study where we use something called the reduced metagenomic sequencing. I think I posted the article for this, and by that technique we can go to the species level. And we looked into all the species in the infant gut, and then we looked into, in a way, looking into for the mother-child, in a way, how in mother-child, for all mother-child pairs, we looked in a ratio of the prevalence. Okay, the prevalence for mother-child pairs versus non-mother-child pairs. So if this bar is above 1, then it will be more prevalent in the mother-child pairs than when the mother and child are not related. And if we do this kind of exercise, we get the Bacteroides genus. This really the 5 top in my mother-child share bacteria. 4 of them belong to Bacteroides, and the 5th belong to Parabacteroides. So these are bacteria that are directly transmitted from mother to child during delivery, and most likely, I can go back to even more like the detailed data in that we really believe that these bacteria have to be like transmitted during delivery, and this, in a way, the mother-child sharing is kept until 6 months of age. So really, if you have a child up to an age of 6 months, you can, in a way, detect, in a way, the higher prevalence of these 4 species, given that the mother has this bacteria, and also given that it has been vaginally delivered. And the blue background here is just the prevalence in the general population. I think is really important results, and by using this kind of more detailed technique that you can go to the species level and even to strain level. I think we have quite good evidence for the direct mother-child transmission for these bacteria. So now I'm going to go into a master project that has digged more into detail in the mother-child transmission. And in this project, we have had access to something fantastically unique through the parental study, where they are taking skin swabs of infants when they are, I think they're 10 minutes old or something like that. So they're swabbed, the children, just after they were delivered, and looking into the bacteria that will be on the skin. And of course, the bacteria that will be on the skin of the child would probably also would be like the bacteria that the child is exposed to when the child is delivered. And I think this is a fantastically unique opportunity to study, in a way, the direct mother-to-child transmission. And what we did, in a way, I can show you this kind of flow chart here. So we took so really the basis of the study was the detection of Bacteroides in meconium, in the first stool sample of the child, and it was the previous results that, in a way, led to that we directly targeted Bacteroides, and we then screened the children in meconium. So we first, in a way, identified about 50 children that were positive for Bacteroides, delivered by vaginal delivery, and then we screened the cesarean section delivered children that were negative for Bacteroides. This is ARPOB gene. It's a gene that is like where we have primers that are specific for Bacteroides. And for cesarean section delivery, we realized that there was very, very low prevalence of this RPB presence. So we could not, in a way, identify children that were positive for RPB. And then we also did quite interesting study that we also identify vaginally delivered children that were RPB negative, in a way. That we could find. We can find in a way vaginally delivered children that were not in a way did not have bacteroides. And then we characterized the species in a way, what kind of bacterial species that were present, and then we match in a way the species between the samples, between the mother, the skin sample, and the meconium sample. We also had a 3-month sample, but this data we have not in a way finalized it. So these are really data that are, this is a really new data. These are data that are going to be written up as a master thesis now in June, and we will also in a way collect all the data that we have, and we will write up a publication on this issue quite soon. If you look at the data, so you have this very colorful data, and I think this is a colorful and very important data. So now we're looking into the meconium, the RPOB positive meconium samples, and we asked the question whether or not we find the bacteria on the skin of the infants and and in the mother. So if we find it on the skin and in the mother also, then the color here will be red, and these are different mother-child pairs. So you can see there are some species that can come up as really red, and especially this Bacillus vulgatus we find highly interesting. So you can see that this stands out as a bacterium that is really distributed across the sample. So it's a mother, in the skin of the infant, and in the meconium. Although we also find it in 3-month samples, but I not included it here because that would be a little bit too complex. But there are also other, in a way, bacterial species that share this kind of property. But this is really some kind of bacterial species that we believe that will be very early transmitted from mother to child. And if you look into these vaginal RPOB negative samples, I think that's really interesting, because these are samples that are screened for meconium. We have not done anything with the skin samples, okay? So we screen them to be negative bacteria in the meconium, the first 2 samples, and we asked the question whether or not they will be negative on the skin samples also. And they are negative on the skin samples. So it seems that although we have a vaginal delivery, these bacteria, very few of them were transmitted. So these are, at least my interpretation would be that these are probably children that have been delivered in a very clean way, in a way resembling that of the C-section. So these are the same. This is the same results for children that have been delivered by C-section. So we really, and then we go for the RPOB negative in meconium, and we look for whether or not we find the bacteria also in skin samples. And we found no of the skin samples. We found none of these bacteria, this bacterial species. We only found it in the mother. In the mothers, the prevalence were more or less equal between these 3 groups. But for me, at least, it seems that in a way it would be the initial seeding that will be really important for whether or not we would inherit this bacterial species. And there will be also, let's say, vaginally delivered children that are potentially delivered in a so clean way that they would reflect what we would see with C-section, cesarean section delivered children. So I'm really eager to go more into this. We have not done the statistics here yet, but I'm really eager when we're going to go into the more statistical analysis on this data. So, and then we also looked into the bifidobacteria. So let's say there are 2 bacteria that we are interested in, and two important in a way genera. This is the Bacteroides and the bifidobacteria. And the bifidobacterium has also in a way been claimed to be more or less directly transmitted from mother to child. But if we go into details in the bifidobacterium transmission, the pattern is completely different from the Bacteroides. So we find it in any case. It's in a way we have in a way the categories vaginal, whether or not it's vaginal delivered or whether or not it's a C-section delivered, and the general pattern would be that the bifidobacteria we found very low level of bifidobacteria in the very early samples, meconium and the skin samples, very low levels. But we started to see, and we have not, I don't know, done the statistics yet, but then we look at, if we look at the 2-month samples, then we start to see potentially some kind of overlap between mother and child in bifidobacterial content. So the interpretation, my interpretation at least, would be that the bifidobacteria probably they are probably mother-child transmitted, but they are transmitted at a later age. They're not directly transmitted. Potentially they're transmitted through breastfeeding or something else, but not at least in the same extent as Bacteroides directly transmitted during delivery. So I think I'm more or less coming to the end of the presentation. So the conclusions, so our conclusions would be that the colonization of the gut starts at the rupture of the amniotic membrane. And that generally, most of the bacteria we get when you're during infancy are inherited from the general population, and it will be the Clostridia that are generated from the general population. The directly mother-child transmitted bacteria would be to a large extent Bacteroides, species belonging to the Bacteroides genus, and especially Bacteroides rugatus. And there could also be direct mother-child transmission of bifidobacteria, but this would happen at a later age than for Bacteroides. So, and then the acknowledgements. So the earlier studies here, the first studies I was presenting is from the NTNU and the PACT study, and the later studies is from UEO and the Preventadal study, and these are like the people that are contributing. Thank you. So hope someone has some kind of questions to me, but very quiet here. So and I hope you got some information from what I told you. Yeah. Thank you.
So thank you very much, Knut. That was really great, very interesting data, and a very nice presentation.
Thank you.
I suggest that you use the chat function to either ask questions or or you can raise your hand, or there are many options. So while you are all thinking, I was wondering, do you have you started looking at, I was interested in the differences between the groups, so who actually got these Bacteroides transmitted and not, and you were speculating about the clean births, etc. But are there other features that you could look into that you would suspect would predispose to transfer or not?
Yeah, so it would be interesting. Of course, so there are a lot. So that's also always the challenge with doing these kind of microbiota studies, is that you have so many options what you want to look for. So I think at least from our, like this study here, it would be like the hypothesis would be like at least for Bacteroides that it will be the direct exposure, and probably then the different. If you can look into delivery in multiple different types of delivery, there's a lot of categories of delivery modes that will be, if you have big enough data sets, you can stratify it on give a better stratification of the delivery modes in a way, to give a more like description on the delivery, labor time, how long, to go more into details on how the delivery occurred, and the potential. But these studies are from Oslo, Østfold, and Stockholm. So they are like in the same regions. It would be very interesting if you could anyway also in the future look into other places. But the challenge here is that really this kind of 10 days, like 10-minute samples are so unique. It's very difficult that you can even get access to this kind of samples. Is because these will be like being there when the child is delivered, that you have to swab the child just once it's delivered. So that will be a really unique sample. So it's a difficult experiment to repeat, at least.
Fully understand. We have a question from Fredrik Müller. Go ahead.
Yeah, hello. Just one question about the Bacteroides, because it seemed that the ability to transfer from mother to child was quite different between the different species in the Bacteroides genus. And do you have some ideas or hypotheses about that?
Yeah. Yes. Because if you look into, I didn't present the data, because we can look into the shift in the Bacteroides with respect to the time until the first. How old the meconium samples are, how old the child, how many hours the child was before we got to the meconium samples. And then we see that in a way that this vulgatus, it seems to be very competitive in the child's gut. It seems to be selected. So it could be, if you go to that's why I showed you the first slide with in a way with this induction of fructose. This is the bacteria theta. I cannot pronounce it, but it is a really common bacterium in the human gut. And this bacterium is able to induce sugar production from the epithelial cells. So it could be that there is some kind of positive selection from the host. For some of the some of the bacteria species, so these are really like selected for by the host. That would be like our initial hypothesis. So these are really bacteria that we would need, and that the host in a way has some kind of selection for those.
Very good. And then we have a question from Mikael.
Hello, thank you for the nice presentation. My name is Mikael, I'm from NOPSC. I'm just, I have a question about, you present the result with the question about in utero colonization, ruptured membrane versus not ruptured membrane. And I think maybe I didn't quite hear what you said, but those with cesarean section and unruptured membrane, they had some copies, bacterial copies. How did you explain those? Was it contamination or?
I would believe it's contamination. So because you never can have anything that's and it could be so. If you take blood samples, you will always find some kind of bacterial DNA. So these are just DNA studies. So you find bacterial DNA everywhere. But the question would be whether or not these are live bacteria. So I would believe that these either will be some kind of trace contamination in our reagents, or it could be bacterial DNA that is present everywhere. Nothing without bacterial DNA. So that it is not, it does not reflect live bacteria. That's my my hypothesis.
Okay, thank you.
So I haven't received any more questions, but I could still I could keep on still. But if you look specifically at Bacteroides and Bifidobacterium, is there any relationship between? And it's a bit strange that they have different ability to transfer during birth. But is it a relationship between the amount of these and the total amount of bacteria that are colonized on the skin, for instance? Have you measured that? What is the skin DNA?
Yeah, so we have in a way. So I haven't gone into details about that, but so there are. We also done studies on the skin DNA, but or the skin microbiota, and also at later ages. But I think Eva, there is another person than me that should in a way address that question. But really, the bifidobacteria, why we address that, because this is selected by mother's milk, and it oligosaccharides in mother's milk. And if you have a 3 to 6 year-old child, that would be really dominating bacteria in the child. So it's really like highly dominant in breastfed children. But Bacteroides is also quite, in a way, it will be the second most dominant bacteria. But these are like, what to say, these are very, very initial results. So we have not like ask me in 10 years, maybe, then I will know more.
Hopefully a bit sooner.
Yes.
Unless you want 10-year follow-up time for the health outcomes. Okay. Anyone else that wants to ask questions now? There's Harald Carlsen. Go ahead.
Hello, Knut. I was a bit curious about this finding that the random mothers and the same mothers, they were, well, the overlap was about the same.
Yeah, if you look at in total, but there are bacteria that are directly mother-child, but in total diversity, this will be hidden by all the bacteria that are not in a way directly mother-child related.
Okay. Did you correct? I mean, if you let's say that you're a child that has been born recently, so the transmission of new bacteria, that is dependent on whether you get around or not.
Yeah, so that it will be
So
more
did
or
you
less.
Characterize the mothers and children? And I mean, if you, let's say that you live in a remote area, then you will probably have very low diversity.
I think that these bacteria will reflect good geographical differences in a way. Well, what's within the general population? I think that would be. So we haven't gone into that kind of studies, looking into the bacteria that are circulating within the general population, in that area. So these are like Scandinavian general population. Potentially you will have more other bacteria if you go to Africa or whatever, and of course you will have that. But these bacteria would reflect most of the bacteria you will inherit them from the general population, not directly from mother to child. That would be our hypothesis. And at least for the Clostridia, which are spore formers, that makes sense. That you inherit them from the general population.
Thank you.
Okay, very good. As there are no further questions, I must say, unless you have some final comment, Marius.
I think this was really great, Knut. So thanks a lot, and thanks for the discussion. And Hanne, do you have any update on the next meeting?
Yep, the next meeting will be more nutrition based. I will send out an email, of course, about this. It's Marit Sinneker will be presenting her HADL model. I'll send out the details there. But also, it's difficult now to keep tabs on the network. So if you have any news, anybody defending their thesis, if you have a position available, just please give me a call or send me an email so we can get the information out. It's difficult when I can't meet anyone. So I would really appreciate it if people contacted me.
Yes, it's actually very important what you say, Hanne, that if there are theses that are defended, we will very much like to put that on the homepage, both so that people could follow the dissertations, but also to keep track of the research that is being made within the network. So that's important.
And articles being published, don't be shy. Just let me know.
Very good.
And also thanks to Hanne for keeping this network alive through Zoom. It's challenging times, and a network is really consisting of the people that meet. So thanks for your work.
Very good, and thank you again, Knut, for this talk. And we have been a lot of participants today, at least 45 at the maximum. So great that so many attended, and I hope to see you again soon in the Zoom meetings, but hopefully also at some time point face to face. So have a nice day and week, and we will meet again soon, hopefully. Okay. Goodbye, everyone.
Bye-bye.
Bye.